You know, with so much going on in the world today, wouldn't it be nice to have a way to just skirt by it? The Phoenix Protocol is all about life extension in a younger body. And the idea that living longer just to live longer is really an unattractive idea. You see, we as people have worked for centuries to try to figure out why we die. And recently, thanks to some researchers, we understand how it is we age and ultimately die. The key to all this is methylation. What happens when DNA is damaged and then after it's repaired, a methyl group is left at the site. It's like putting a roadblock on a highway. The messenger RNA can't get past the roadblock. And even though the DNA has been repaired, and here's the irony, it can't be used as long as a roadblock is there. So what we're going to talk about today is sirtuins, because sirtuins are the way the body removes the roadblocks. And another molecule, NAD+, which is what activates sirtuins. One of the reasons we have this illness currently going on is because as you age, the amount of NAD+ decreases over time for a variety of reasons, and I'll show you how to raise those levels because you need NAD+ to activate the enzymes that identifies DNA damage, that activates the enzymes that removes the methylation, and activates the sirtuins that do this work, especially in the cytoplasm. The idea here is that there's two forms of tagging for genetics. There's acetylation, where an acetyl group is put on histone ends, and methylation, where methyl groups are put on DNA themselves. Now, methylation and acetylation is not a bad thing when it's done correctly because the body uses methylation in all the different types of body cells to identify which proteins can be made in those cells and which proteins should not be made in those cells by silencing genes. And it's those methyl tags that start the process of creating proteins that cell needs to remain healthy, be it an eye cell or a liver cell. The problem comes, like I've said, when DNA is damaged and the cell has to repair itself. And when it does that, an enzyme called PARP is attracted to the site, is activated by NAD+, signals which base excision repair enzyme is needed for a single strand, double strand, code violation, what have you. And during that process, a methyl group is left at the repair site. When you have enough NAD+ to activate Sirtuin, Sirtuin can come in and remove the methylation tag, and the DNA can then rewrap around the histone correctly, and that allows that code to be accessed again to make a protein you need. So when we look at DNA under the microscope, DNA is assembled into a series of histones. These are nucleosomes that wrap up the DNA, these little spools, and these spools allow the DNA to be either accessible or not accessible, silenced or active. The DNA can be methylated and acetylated on the cytosine amino acid. It can be methylated, it can be acetylated, it can be phosphorylated. And these things allow DNA to be either read or not read. So when we look at what happens in the cells themselves, we see that the cells are just basically molecular machines. And these molecular machines are activated by start and stop signals, methylation, acetylation. And when the proteins have been made, those tags need to be taken off. And this is similar to what happens in a cell when a virus attacks. When a virus enters the cell, it starts affecting the calcium pumps, and the calcium levels inside the cell are interrupted. When this happens, calcium levels are raised. It turns on acetyltransferase, which starts putting acetyl groups on cytosine in the cell. This affects the function of the cell. But in particular, because these are RNA viruses, it starts tagging the RNA virus code to be able to start and stop its main sequence replication and the subgenomic replications to make the shell and the protein spikes and all that stuff. If Sirt2 has enough NAD+, Sirt2 can take those tags right off. The virus never gets replicated, the shells and protein spikes never get replicated, and those parts can be moved to lysosomes for degradation, where they're dissolved back down into amino acids and then used again to make more cells. The key to longevity and life in a younger body is to remove these tags when they're not needed. So if you really want to live longer and you really want to be healthier, you need to remove that stuff. And you can do it in a lot of ways. You can take the Physitin before you fast, which removes a lot of senescent cells before you fast. It makes it easier for your body to remove senescent cells. But again, why do the senescent cells become senescent? Because they've been so damaged they can't make proteins by methylation tagging and acetylation tagging. Everything in the body is made from proteins accessed from the histone libraries in each cell. DNA is wrapped around histones to silence some proteins while making others available. This prevents proteins in an eye not being made in the liver. Remarkably, each cell has its sirtuin codes readable in every cell. When the histones are acetylated to open up code to transcribe a sirtuin, messenger RNA finds the correct methyl tag on the DNA to make the cell-specific protein needed to begin that code, and happily chugs along and manufactures the various 7 different sirtuins for the nucleus, mitochondria, and cytoplasm. The sirtuins are very complex molecules, and each has a domain or a pocket that is a docking location to become activated by another molecule, NAD+, to perform their work. To demethylate DNA after repair or deacetylate the DNA after transcription. In fact, when DNA has been tagged with a methyl group, NAD+ activated sirtuins are drawn to the site to remove the tag, allowing the code to be tightly rewrapped onto the histone to protect it until it's needed again. Methylation is a necessary function of cells to identify where to open up the DNA to read a code. Problems occur when the DNA is damaged. When this happens, another NAD+ activated molecule, PARP, is drawn to the damaged site, where it affixes itself to the damaged section and signals the type of base excision repair to fix the damage. Single-strand breaks, double-strand breaks, base insertion violations, any number of issues. Often a methyl tag or acetyl tag is left at the site, preventing the histone from winding the code into a safe spool once again. The keystone in this arch of DNA protection is NAD+, because without it, sirtuins and PARP, as well as the protection of ATP and mitochondria, can't do their job. The DNA is not repaired and the cell goes down the road to senescence. And it's a very important molecule. It's used in brain and nervous system sensory and motor function, in the liver for gluconeogenesis and fatty acid oxidation, in the vascular system for endothelial function, the interior lining of the arterial walls, a heart, its cardioprotection, lymphoid tissue, immunity and anti-inflammatory, the reproductive organs for fertility, and the kidneys for renal protection, pancreas for insulin secretion, muscle, insulin sensitivity and fatty acid oxidation, adipose tissue for lipogenesis. Exercise, diet, and NAD boosters increase NAD+. The way NAD+ connects to all this functionality is by its activation of the 7 sirtuins. Sirt1, 3, 6, and 7 are used for facultative heterochromatins to regulate and protect the chromosomes and the DNA wrapped around the histone. Constitutive heterochromatins for Sirt1 and Sirt6, cell cycle checkpoints, Sirt1 and Sirt2, cell fate control, Sirt1, 2, 3, 6, and 7, DNA repair, Sirt1 and Sirt6, DSB repair, Sirt1, Sirt6, Sirt1, Sirt3, Sirt6, and of course mitochondrial function, which is Sirt3 and Sirt4, with some use of Sirt1. In fact, the sirtuins are very essential for circadian metabolic output, basically regulating what happens during a 24-hour period. Sirt3 regulates mitochondrial enzymes, Sirt6 regulates circadian genes, and Sirt1 regulates circadian genes as well, and the production of NAMPT, which is part of the pathway to make NAD+. Optimal mitochondrial function is regulated by NAD+, activated Sirt1 and Sirt3, and critical for mitochondrial biogenesis, making more mitochondria. Upregulation in various pathways assure NAD+ is sufficient to maintain mitochondrial function. It supports chromosome stability, DNA repair with part 1, immune cell signaling, energy enzyme activity, longer telomeres, neurotransmitters and brain health, longevity mechanisms, Sirt1 through 7, and energy production of ATP. But sadly, there is a reduction of NAD levels over time that directly affects aging because without it, damaged DNA methylation is not removed, resulting in less access to proteins to remain young. Age is a mechanical issue. It's not a chronological one. The direct effects of this reduction of NAD+ with age is that when Sirt2 in the cytoplasm is inactivated, acetyl tags placed on the genome of virus can't be removed to prevent replication, and viral infection can't be stopped. So how do we raise levels for not only health but for longevity? Because the Phoenix protocol is designed to radically extend lifespan by keeping these NAD+ levels high as an essential part of functional immortality. We know for sure that resveratrol and exercise has a positive effect on NAD+ levels, but we know for certain that dry fasting activates NAD synthesis. There are 3 biochemical pathways because this is such an important part of homeostasis. The NAD+ molecule is made from the amino acid L-tryptophan in the de novo synthesis pathway, and the Preece-Handler pathway using flush niacin, nicotinic acid. It's also produced in a salvage pathway using the non-flush niacin, nicotinamide riboside, and NMN, as well as resveratrol. But the best way is to incorporate weight and strength training, resveratrol and nicotinamide riboside along with NMN, as well as fasting, because it's been proven that dry fasting upregulates the synthesis of NAD+. So now you know it's really important to have NAD+ in your body at all times. You have to activate your sirtuins. Now, there's obviously several ways you can do this. My personal choice is to use the de novo pathway and the salvage pathway in unique ways. First of all, we know that resveratrol has a 5-fold increase in stimulating the production of NAD+. So what I use is the resveratrol to work on the salvage pathway to synthesize more NAD+, and I work on the de novo pathway with tryptophan and niacin. So the easy way to do this is just take tryptophan, niacin, resveratrol, transresveratrol, and work both sides of the ability to synthesize the NAD+. You see, this is all part of an integrated program in the Phoenix protocol. The book talks about it extensively, and you can pick this up on Amazon. But the point is, in the book, it's part of a systematic way to achieve longevity. The Phoenix protocol is certainly about dry fasting and healing and rapid healing because that's what it does. It's the best at that. But it's not specifically for fasting. The Phoenix protocol is for life extension, radical life extension. And the only way to do that is to make sure that you are demethylated and deacetylated. So the only place you'll find those in the molecules inside your cells are in the right places. So you can access all the codes. Because youth is not measured in age, youth is measured in functionality. So I'll see you next time.